Welcome to the Antigen Recognition by Lymphocytes module. And in this talk, we're going to learn about different structural aspects of the B cell receptor. We're going to first learn about the different types of immunoglobulins and the structure of the B cell receptor. We will also learn how to identify the immunoglobulin fold and the hypervariable or CDR regions. Finally, we will also learn about the different forces involved in binding of antigens to antibodies or immunoglobulins. Let us begin with a summary of innate versus adaptive immunity. As we learned previously, innate immunity is quick, is fast, and is the first defense that plays a role in controlling pathogens that exhibit certain molecular patterns. However, innate immunity a lot of times is not enough for the efficient removal of a wide range of pathogens, and for that, an adaptive immune response is required. The part of the adaptive immune response are the lymphocytes, which are able to recognize a wide variety of antigens that are present on all kinds of microorganisms. Just to summarize, an antigen is a molecule or part of a molecule that is recognized by special recognition proteins on these lymphocytes. And in the case of a B lymphocyte, this special recognition protein is the B cell receptor, while in the T lymphocyte, this special recognition protein is the T cell receptor. Let us first focus on the B cell receptor or the immunoglobulins. Now do remember that antibodies and immunoglobulins often refer to the same kind of molecule. There are 5 classes of immunoglobulins that our bodies make, and they're all listed in this picture below. So we have the IgG, IgE, IgD, and the Ig in this case stands for immunoglobulin. We also have multimeric immunoglobulins like IgM and IgA. If you focus on the black box, you can see that IgM is made up of 5 immunoglobulins that are linked together, and hence it is a pentameric structure. While in IgA, we have 2 antibodies that are bound together to form a dimeric structure. All of these immunoglobulins have their own specific functions and features that we will learn as we go through the course. Let us look at the basic structure of the antibody. Now, each antibody is made up of 2 different but identical kinds of polypeptides. If you focus on the figure, we can see this Y-shaped structure that is formed by the antibody. Let us look at the different parts of it. So there are 2 heavy chains, and they have been outlined in those black structures, and they are the longer lines that we can see. Every antibody will also have 2 light chains that are denoted in the figure right now. Now, when we focus on those heavy chains, those 2 heavy chains that are part of the antibody are identical to each other, and they are linked together by disulfide bonds that keeps them closer to each other. And the disulfide bond is shown there in that smaller box. Now, each heavy chain is linked to a light chain, as shown in the figure, and that also occurs through a disulfide bond. So right now we're looking to the left side of the figure where we have 1 heavy chain bond to 1 light chain. And the same thing is happening on the other side of the antibody too. And now we can see in the smaller boxes where the disulfide bonds are present that are linking each heavy chain to a light chain. These disulfide bonds help the antibody stay intact and not fall apart. So as you can see, every antibody is made up of 4 polypeptides, which are 2 heavy chains and 2 light chains. All immunoglobulin domains have a similar 3-dimensional structure, and they're rich in beta strands. So both the V and C domain of the heavy chain as well as the light chain is made up of 2 beta sheets. These beta sheets fold to form a beta sandwich, and if you focus on the figure that's shown in the black box, we can see the 2 beta sheets. So here's the first one that you can observe that is formed by the yellow colored beta strands, and the second beta sheet is the one that is formed by the green colored beta strands. These fold with each other to form the beta sandwich and are kept intact through a disulfide bond. And this folded structure is called as the immunoglobulin fold. And the similar immunoglobulin fold can also be seen in the light chain's variable domain as well as in the heavy chain's V as well as C domains. If we take a closer look at the beta strands of the immunoglobulin domains, there is a characteristic arrangement of these beta strands. You are not expected to remember the directionality of the beta strands. However, it is good to note that when we look at the V domain, it tends to have additional beta strands than the C domain. When we look at the amino acid sequences in the V region of the immunoglobulin, so this is the amino terminal region of the heavy chain as well as light chain, and is showed in blue and red colors in the figure, there is a lot of variation in specific parts of this V region. And that's what's shown in this figure. So if we look at the figure in the black box, we're looking at the V region of the heavy chain. And on the x-axis of that plot, we can see the different amino acid numbers. And on the y-axis, we're looking at the variability in the amino acid sequence. And what we can see is that there are 3 regions where there is a lot of variability in the amino acid sequence of the V region. And these 3 regions where there's a lot of variation are called as hypervariable regions. Now this is seen in the V region of the heavy chain, but it is also seen in the V region of the light chain. So here also we can see that there are 3 regions where there is a lot of variability in the amino acid sequence. The regions between these hypervariable regions, which are shown in blue or yellow in the case of the heavy chain and the light chain respectively, are called as the framework regions. Now, where do these hypervariable regions lie when we look at the 2-dimensional structure of the immunoglobulin? When we look at the 2-dimensional structure of that V domain of the immunoglobulins, we can see that those hypervariable regions are actually formed in these loop-like structures, and they are shown in red in that black box shown in the figure above. When we look at it in the 3-dimensional folded structure, here we can actually see that they are also present in those loop-like structure, and these loops actually form the antigen binding site of the immunoglobulin. The hypervariable loops on an antibody or immunoglobulin have a special name, which are called as the complementarity determining regions, or CDRs. Now, each V region of the heavy chain as well as the light chain has 3 CDRs, or 3 complementarity determining regions. Now, let us see what is the importance of these CDRs. Different antibodies will have different amino acid sequences in their CDRs. And these differences in the amino acid sequence influences the shape and the properties of the CDRs in one antibody as compared to, let's say, another antibody. These differences in the shapes and properties of the CDRs allows one antibody to bind and recognize one type of antigen, whereas another antibody can now recognize a different type of antigen. And that is what is shown in this figure, where depending on the shape and properties of the CDRs, this specific antibody is able to bind to, let's say, an antigen that is circular. However, in this case, we are seeing that it is able to bind an antigen that looks different. And in this case, we can now once again see that the shape and the property of the complementarity determining region is different from the previous ones that we saw, and hence it is able to bind to an antigen that looks different than the others. And here we have another example of the same concept. Thus, the shape and properties of CDRs are the ones that will determine the kind of antigen that that antibody will bind to. It should be noted that even though we say that antibodies are able to recognize and bind to antigens, they do not recognize the entire structure of that antigen. They will actually bind to a small region that is present on the surface of that antigen, and this small region is called as an epitope. Many antigens are proteins, but please do remember that not all antigens are proteins. We can have antigens that are carbohydrates. We can even have antigens that are lipid-based, and thus there is a wide variety of antigens. When we look at different types of epitopes, there are two main types. The first one is where the epitope is linear or continuous, and in this case, the immunoglobulin or antibody is recognizing the basic sequence of a linear epitope. An example of this would be, for example, a protein that is an antigen, and we're looking at the linear amino acid sequence, where the amino acid sequences are right one after another in the epitope. Another example of an epitope is a conformational or discontinuous epitope. And in this case, what the antibody is actually recognizing is a structure formed by that epitope. So if we look at our protein example, in this case, the immunoglobulin or antibody is not recognizing that linear sequence of amino acids that are right one after another, but it is actually looking at a conformational shape that is being formed by amino acids that are not necessarily near each other. And thus we can have conformational or discontinuous epitopes. Antigens are able to bind to antibodies, or the epitopes in the antigens are able to bind to antibodies through non-covalent interactions. In this table, we can see different types of non-covalent forces that are involved when an antigen binds to an antibody. They can be polar in nature, like electrostatic forces, or they can be non-polar in nature, like the van der Waals or hydrophobic forces. When we look at the binding of an epitope to an antigen, we can have either one type of a non-covalent force or a combination of forces that are listed in this table. Let us now look at another region of the immunoglobulins that's called as the Hinge region. Now the Hinge region is the region between the Fab and the Fc region of the immunoglobulin. The Hinge region that's shown in the box is important because it provides the immunoglobulin with flexibility and allows many immunoglobulins to bind to multiple antigens. So here's an example in the figure to the left, that's right below the text, where we can see 3 immunoglobulins are binding to 3 antigens. And the way the binding has occurred, the angle between the different arms of the immunoglobulins is 60 degrees. However, when you look at the figure right next to it to the right, we can see 4 immunoglobulin molecules that are now binding to 4 antigens that are near each other. And in this case, when you look at the angle between the arms, it is 90 degrees. This change in the angle from 60 degrees to 90 degrees is due to the flexibility that is provided by the Hinge region. Thus, the Hinge region plays an important role in providing flexibility to the immunoglobulin when it binds to antigens. So far, we have looked at the antibody structure of antibodies or immunoglobulins that are found in humans. And as shown in that black box, we can see that there are 2 heavy chains and 2 light chains. And each heavy chain and light chain will have a variable domain, as well as they can have constant domains or C domains. In the case of the heavy chain, we can have either 3 or 4 C domains. Now, when we look at the antibody structures of other organisms, there can be differences in their structure. However, the function is the same, which is to recognize epitopes that are present on antigens and to bind to them. Let's look at the camelid immunoglobulin, and in this case, what we can see is that there are no light chains. So all we have are 2 heavy chains that are bound to each other. When we start looking at the shark immunoglobulin, once again, we don't see any light chains. There are 2 heavy chains that are bound together. However, when we look at the number of domains that are present on those heavy chains, we can see that there are many more domains present than what is there in the human. In spite of these differences, as I said before, the function is the same for all of these immunoglobulins. As we go in our course, we will focus more and more on human antibodies or immunoglobulins. So with this, we come to the end of the B cell receptor, which is also called as an immunoglobulin, and when it's secreted is also called as an antibody. With this, we come to the end of our talk where we learned about the different immunoglobulins and the structure of the B cell receptor, which includes the immunoglobulin fold. We also learned about hypervariable or CDR regions that are found in the B cell receptor and the different forces involved in the binding of the antigen to the antibody or immunoglobulin.